नेक्स्ट है मसल फिजियोलॉजी। तो इसमें देखेंगे राइट शॉर्ट नोट ऑन द सार्कोमियर, ये राइट ब्रीफली ऑन द सार्कोमियर। तो sarcomere is the functional unit of the skeletal muscle. Each sarcomere extends between the two jet lines of the myofibril. Each myofibril consists of the alternative A band or the dark band and the I band or the light band. In the middle of the A band, there is a light area called the H zone, and in the middle of the H zone lie the middle part of the myosin filament, which is called the M line. I band is divided into two equal halves by the means of the jet line. The part of the myofibril between the two jet lines is known as the sarcomere. Sarcomere consists of the two types of the myofilaments, actin filament and the myosin filament. Actin filament are the thin which extend from the either side of the jet line and run across the I band. इसके बाद फिर आता है myosin filament are the thick which are situated in the A band. These are from lateral processes or cross bridge arising from the myosin filament, which are enlarged structure and are called the myosin head, which are at the tip of these bridges. These myosin head attach themselves to the actin filament and push the actin or the myosin filament during the muscle contraction by the means of the mechanism called the sliding or the ratchet mechanism. During the muscle contraction, actin filament glide towards the M line or the H zone, and the I band are shortened or disappear during the contraction of the muscle. During the relaxation, the jet line and the actin filament come to the original position. Actin and the myosin filaments are formed by the contractile protein, out of which myosin is formed by the myosin protein and actin is formed by the actin, troponin and the tropomyosin protein. यहाँ पर एक सार्कोमियर दिखाया हुआ है, इसमें एच जोन हो गया, ए बैंड, आई बैंड, जेड लाइन, मायोसिन फिलामेंट, एक्टिन फिलामेंट और दिया हुआ है एम लाइन। इसके बाद फिर आता है डिस्क्राइब द जनरेशन ऑफ द एक्शन पोटेंशियल इन द नर्व। ये राइट अ शॉर्ट नोट ऑन द नर्व एक्शन पोटेंशियल, राइट अ शॉर्ट नोट ऑन द एक्शन पोटेंशियल। So when the nerve is stimulated, a series of the changes occur in the membrane potential, which is called as the action potential. The action potential occurs in the two phases, depolarization or repolarization. Depolarization, when the impulse reaches the nerve, the polarized condition minus 70 millivolt is altered. That means the resting membrane potential. और इसके बाद फिर आता है repolarization. Within a short time, the nerve obtains the resting electrical potential once again. Interior of the nerve becomes negative and exterior positive. So the polarized state of the nerve is re-established. This process is called as the repolarization. Resting membrane potential recorded at the straight baseline of the 20 millivolt. When a stimulus is applied, there is a slight irregular deflection of the baseline for a very short period. This is called as the stimulus antifact. Latent period. The stimulus antifact is followed by a short period without any change. That means a latent period. Firing level. The depolarization starts up to the latent period. Initially, it is very slow up to the minus 55 millivolt. Then the rate of the depolarization increases suddenly. The point at which the rate of depolarization increases is called as the firing level. From the firing level, the curve reaches isoelectric potential rapidly and then overshoots zero line up to the plus 35 millivolt. A spike potential. When the depolarization is completed, plus 35 millivolt, the repolarization starts with a rapid increase in the depolarization and rapid decrease in the repolarization are together called a spike potential. इसके बाद फिर आता है the rapid fall in the spike potential is followed by the slow repolarization process. This is called as the depolarization. After reaching the resting level minus 20 millivolt, it becomes little more negative than the resting level. In above bay, the action potential is generated, propagated and produced along the curve. तो यहाँ पर एक्शन पोटेंशियल दिखाया हुआ है और एम्पलीट्यूड मिलीवोल्ट में, स्टिमुलस आर्टिफैक्ट, लेटेंट आर्टिफैक्ट, फायरिंग लेवल, आफ्टर डिपोलाइजेशन, आफ्टर हाइपरपोलाइजेशन है जो दिखाया हुआ है। नेक्स्ट है डिस्क्राइब द मैकेनिज्म ऑफ द एक्साइटेशन, कॉन्ट्रैक्शन, कपलिंग इन द मसल। या राइट ब्रीफली excitation, contraction, coupling in the skeletal muscle. So when the muscle is stimulated, it is excited and action potential is developed. Then the muscle starts contracting. The contraction of the muscle starts due to the pulling of the actin filament by the cross bridge from the myosin filament. The process involved in between the excitation and the contraction of the muscle is called the excitation contraction coupling. When the impulse through a motor neuron reaches the neuromuscular junction, acetylcholine is released. Acetylcholine causes the opening of the ligand-gated sodium channel so that sodium ions enter the neuromuscular junction. This led to the development of the end plate potential. The end plate potential causes the generation of the action potential from the muscle fiber. The action potential spreads over the sarcolemma and also into the muscle fiber through the T tubules. When the action potential reaches the cisternae of the L tubules, these cisternae are released into the sarcoplasm. The calcium ions from the sarcoplasm move towards the actin filament to produce the contraction. Thus, the calcium ions are linking or coupling materials between the excitation and the contractions of the muscle. Hence, the calcium ions are said to form the basis of the excitation contraction coupling. Next है energy for the muscular contraction. The energy for the movement of the myosin head power stroke is obtained by the breakdown of the adenosine triphosphate ATP. Write briefly on sliding filament theory of the muscle contraction of skeletal. This theory explains the sliding of the filament is brought about by the repeated cycle of the formation of the cross bridges between the head of the myosin and the actin molecules. A step of the cross bridge for cycling. So initiation of the cross bridge cycling. At the time of the rifting stage, troponin 1 is lightly bound to the actin and tropomyosin molecules are located inside the groove between the strands of the actin filament in such a manner that they block the myosin binding site on the actin. So during the rifting stage, no actin-myosin cross bridge are formed. Thus, the tropomyosin complex called the reflexing protein inhibit the interaction between the actin and the myosin. When the activation takes place, the calcium ions will lead in the cytosol from the terminal effuston of the sarcoplasmic reticulum and get attached to the troponin C subunit of the protein troponin. It results in change which causes the tropomyosin molecule to move laterally, uncovering the binding site on the actin molecule for the head of the myosin molecule. 7 myosin binding sites on the actin filament are uncovered for each molecule of the troponin that binds the calcium ion. Thus, the cross-bridge cycle initiated by the lateral movement of the tropomyosin. नेक्स्ट फिर आता है फॉर्मेशन ऑफ द एक्टिन मायोसिन कॉम्प्लेक्स. तो हेड ऑफ द मायोसिन मॉलिक्यूल bonds with the adenosine triphosphate. The ATPase activity of the myosin head immediately cause the breaking of the ATP to the adenosine diphosphate ADP and the phosphate cleavage product which remain bound to the myosin head. The head of the myosin becomes energized. Activated myosin head extends perpendicularly towards the actin filament and gets attached to the actin filament. Formation of the actin myosin ADP phosphate complex leads to the following events: release of the phosphate and ADP from the complex, a conformational change in the myosin head causing it to flex towards the arm of the cross bridge, the flexion of the myosin head from the high energy 90-degree conformation to the low energy 45-degree conformation generates the mechanical force, the power of torque. Next, detachment of the head of the cross bridge from the active site of an actin filament, release of the ADP and the PI phosphate allow a fresh ATP molecule to bind the myosin head. Myosin ATP complex has a low affinity for the actin and therefore it results in the dissociation of the myosin head from the actin filament. Reactivations of the myosin head. तो freshly bound ATP molecule split again and myosin head is reactivated for the next cycle to begin. So with each cross bridge cycle there is movement of the actin filament towards the center of the myosin to a small degree. Repeated cross bridge cycling cause the movement of the actin filament of either side towards the center of the myosin filament of the sarcomere leading to the muscle contraction. अगला क्वेश्चन है, राइट अ शॉर्ट नोट ऑन द मिस्थेनिया ग्रेविस. तो मिस्थेनिया ग्रेविस इज सीरियस एंड समटाइम्स अ फैटल डिजीज इन विच द skeletal muscle are weak and tired easily. It is caused by the formation of the circulating antibody due to the nicotinic acetylcholic receptors. These antibody destroy some of the receptors and bind other to the neighboring receptors, triggering their removal by the endocytosis. The reason for the development of the autoimmunity to acetylcholine receptor in this disease is still unknown. Another condition which resembles to the myasthenia gravis is the Lambert-Eaton syndrome. Write a short note on the tetany. It is a condition characterized by the hyperexcitability of the nerves and the skeletal muscle, resulting in the muscular spasm, particularly in the feet and the hand. The increased neural excitability results in the convulsion. There are two types of the tetany. Hypocalcemic tetany, if the plasma calcium level falls below 6 mg/% from its normal value of the 9.4 mg/%, the hypocalcemic tetany occurs. और नेक्स्ट में आता है फिर यहाँ पर दिया स्लाइडिंग फिलामेंट थ्योरी ऑफ द मसल कॉन्ट्रैक्शन. तो ये दिया हुआ है मायोसिन क्रॉस ब्रिज अटैचेड टू द एक्टिन फिलामेंट, मायोसिन हेड हाई एनर्जी कॉन्फ़िगरेशन एडीपी और फॉस्फेट. नेक्स्ट है वर्किंग ऑफ स्ट्रोक. द मायोसिन हेड pivots and bends as it pulls on the actin filament, sliding it toward the M line. तो यहाँ पर ADP and the PI दिया हुआ है, inorganic phosphate released. नेक्स्ट है as the new ATP attached to the myosin head, the cross bridge detaches. तो यहाँ पर myosin head लो एनर्जी कॉन्फ़िगरेशन दिया हुआ है और ATP दिखा रहा है. फिर as the ATP is split into the ADP and the PI, cocking of the myosin head occurs. यहाँ पर thick filament दिया हुआ है और ATP hydrolysis ये दिखा रहा है. So the signs and the symptoms of the hypocalcemic tetany are the carpopedal spasm, laryngeal stridor, that means the contractions of the laryngeal muscle, dilatations of the heart, decreased permeability of the cell membrane. Latent tetany, hypocalcemia causes the hyperexcitability even before onset of the tetany. It is called as a latent tetany. It is characterized by the general weakness and the cramps in the feet and the hand. अगला क्वेश्चन है, डिस्क्राइब द स्ट्रक्चर एंड द फंक्शन ऑफ द स्केलेटल मसल. एड ए नोट ऑन द मोलिकुलर मैकेनिज्म ऑफ द मसल कॉन्ट्रैक्शन एंड द न्यूरोमस्कुलर ट्रांसमिशन. डिस्क्राइब द स्ट्रक्चर ऑफ द स्केलेटल मसल एंड द मैकेनिज्म ऑफ द मसल कॉन्ट्रैक्शन. तो स्ट्रक्चर ऑफ द स्केलेटल मसल कंसिस्ट ऑफ द 1 लाख मसल्स ऑफ द or 1 lakh of the muscle cells that are also known as the muscle fibers, or these cells act together to perform the functions of the a specific muscle of which they are a part. Periosteum is the outer layer of the bone. It is to this layer that ligaments and tendons are attached. Tendon attach muscle to the bone. They are tough, pale colored vertex cord formed from many parallel bundles of the collagen fiber. Tendons are flexible. They bend around the other tissue, changing position as they move, yet inelastic. Fascia. The word fascia means bandage. A fitting analogy as the tissue called the fascia takes the form of the feet or broad bands of the fibrous connective tissue that cover the muscle or the organ, forming an outer wrapping. There are two types of the fascia, superficial fascia or deep fascia. Superficial fascia consists of the areolar connective tissue and the adipose tissue and may also be referred to as the subcutaneous layer of the skin. Deep fascia is more relevant to the study of the muscle structure because it is deep fascia that holds the muscle together. It consists of the dense fibrous connective tissue. Next, skeletal muscle, voluntary muscle. So the type of the muscle that cause the movement of the skeletal system, especially limbs and of the skin in the case of the muscles of the facial expression in the head and the neck area has many names. These include the skeletal muscle because it moves bones, voluntary muscle because it is usually under conscious control, and striated muscle because they have a stripped appearance. Perimysium is a fibrous sheath that surrounds and protects the bundles of the muscle fiber. Epimysium is a fibrous elastic tissue that surrounds the muscle. फिर आता है fascicle. The term fascicle, sometimes expressed as the fasciculus, refers to the bundle such as a bundle of the muscle fiber, example as illustrated above, or the alternatively a bundle of the nerve fiber. Endomysium is the name of the fine connective tissue sheath that surrounds or covers each single individual muscle fiber. Muscle fiber, a muscle cell. Muscle fiber, also known as the muscle cell, are special cells that are able to contract, thereby causing the movement of the other tissue parts of the body. There are the three types of the muscle: a striated, a skeletal muscle causing the movement of the bone or limbs, a smooth muscle surrounding the organs and the blood vessel, and the cardiac muscle forming the walls of the heart. फिर आता है myofibril. Myofibrils are the small contractile filaments located within the cytoplasm of the striated muscle cell. These filaments cause the distinctive appearance of the skeletal, voluntary, or striated muscle because they consist of the bands of the alternating high and the low refractive index. These give the muscle their stripped appearance. फिर आता है functions of the skeletal muscle. The skeletal muscle are the mechanism for the powering human movement, while individual muscles are typically regarded as a distinct organic structure. The skeletal muscle are the largest organ grouping in the body. The skin is the largest contiguous organ. Virtually all joints are moved by the pairs of the muscle working in a contrasting but complementary way. One set providing the extension of the joint extensor, the opposing or the antagonist set containing the with flexion or blending capability. The skeletal muscle perform the important roles relative to the body energy system while the body is fasting or otherwise not ingesting food to be converted into the useful energy sources. The skeletal muscle release amino acids during the period of the fasting, particularly alanine and the glutamine. These acids work in the bloodstream to maintain the body blood's glucose level, stimulating the conversion of the glycogen stored in the liver into the glucose. A skeletal muscle moves the skeleton and is responsible for all our voluntary movement as well as for the automatic movement required. For example, to stand, to hold up our head and to breathe. A skeletal muscle maintains the heat regulation. अब यहाँ पर दिखाया हुआ है स्केलेटल मसल फाइबर, इसमें दिखाया हुआ है मायोफिब्रिल, मसल फाइबर सेल, इंडोमाइसियम, फैसिकल, इपीमाइसियम, पेरीमाइसियम, स्केलेटल, वॉलेंट्री मसल, deep fascia, tissue covering the muscle sewn cut back from the muscle for the clarity of distribution. फिर है tendon attaching the muscle to the bone और periosteum layer of the bone दिखाया हुआ है. Next है molecular mechanism of the muscle contraction. तो molecular mechanism of the muscle contraction is explained on the basis of the sliding filament mechanism. Sliding filament theory explains that sliding of the filament is brought about by the repeated cycle of the formation of the cross bridge between the head of the myosin and the actin molecules. Steps of the cross bridge cycling. In sequence of the cross bridge cycling, at the time of the rifting stage, troponin 1 is lightly bound to the actin and the tropomyosin molecule are located inside the grooves between the strands of the actin filament in such a manner that they block the myosin binding site on the actin. So during the rifting stage, no actin-myosin cross bridge are formed. Thus the tropomyosin complex, so-called relaxing proteins, inhibit the interaction between the actin and the myosin. When the activation takes place, the calcium ions released in the cytosol from the terminal system of the sarcoplasmic reticulum and they get attached to the troponin C subunit of the protein of troponin. It results in a change which causes the tropomyosin molecule to move laterally, uncovering the binding site on the actin molecule for the head of the myosin molecule. 7 myosin binding site on the actin filament are uncovered for each molecule of the troponin that binds the calcium ion. Thus the cross bridge cycle is initiated by the lateral movement of the tropomyosin. अगला है formations of the actin-myosin complex. The head of the myosin molecule binds with the adenosine triphosphate. The ATPase activity of the myosin head immediately cause the breaking of the ATP to adenosine diphosphate (ADP) and the phosphate cleavage products which remain bound to the myosin head. The head of the myosin becomes energized. Activated myosin head extends perpendicularly towards the actin filament and gets attached to the actin filament. Formation of the actin myosin ADP phosphate complex lead to the following events: release of the phosphate and ATP from the complex, a conformational change in the myosin head causing it to flex towards the arm of the cross bridge, the flexion of the myosin head from the high energy 90-degree conformational to the low energy 45-degree conformation generates the mechanical force, the power of stroke. फिर आता है detachment of the head of the cross bridge from the active site to an active filament. Release of the ADP and the phosphate allow a fresh ATP molecule to bind the myosin head. Myosin ATP complex has low affinity for the actin and therefore it results in a dissociation of the myosin head from the actin filament. Reactivation of the myosin head. Freshly bound ATP molecule splits again and myosin head is reactivated for the next cycle to begin. So with each cross bridge cycle, there is movement of the actin filament towards the center of the myosin to a small degree. Repeated cross-bridge cycling cause movement of the actin filament of the other side towards the center of the myosin filament of the sarcomere leading to the muscle contraction. अब यहाँ पर द न्यूरोमस्कुलर जंक्शन दिखाया हुआ है. इसमें हो गया एक्शंस ऑफ द मोटर न्यूरॉन, माइलिन शीथ, एक्शन पोटेंशियल, फिर एक्शन टर्मिनल बटन, वेसिकल्स ऑफ द एसिटाइलकोलिन, वोल्टेज गेटेड सोडियम चैनल, प्लाज्मा मेम्ब्रेन ऑफ द मसल फाइबर, एसिटाइलकोलिन रिसेप्टर फैट, एसिटाइलकोलिन स्टेज, न्यूरोट्रांसमीटर गेटेड चैनल. फिर यहाँ पर मसल फाइबर, एक्जन टर्मिनल्स, वोल्टेज गेटेड कैल्शियम चैनल, एक्शन पोटेंशियल प्रोपगेशन इन द मसल फाइबर, ये दिखाए हुए हैं. इसके आगे फिर आता है न्यूरोमस्कुलर ट्रांसमिशन. न्यूरोमस्कुलर ट्रांसमिशन इज डिफाइंड एज द रिलीज ऑफ द एसिटाइलकोलिन फ्रॉम द नर्व एंडिंग एंड एक्टिवेशन ऑफ द रिसेप्टर इन द मसल एंड प्लेट. मैकेनिज्म ऑफ द ट्रांसमिशन: नर्व एक्शन पोटेंशियल, nerve axon action potential arrives at the end plate transiently, depolarizing the axon terminal and then dying out. Axon terminal depolarization opens calcium ion channels briefly, permitting the calcium to diffuse into the axon terminal. Increased intercellular calcium leads to the exophytosis of the sum of the acetylcholine containing vesicle, which discharge their acetylcholine into the intercellular cleft. FCH diffuse to the muscle fiber end plate region. Diffusion is rapid because the distance is short. FCH attaches to the binding site on the muscle fiber, FCH receptor, binding site on the non-specific small ion channel and causes the channels to open. Na plus diffuse into the muscle cell, causing the end plate depolarization. The depolarization is termed as the end plate potential EPP. EPP is local, non-propagated depolarization. No action potential is generated by the FCH sensitive channel since they are not voltage sensitive. इसके बाद फिर आता है end plate potential spreads passively to the nearby muscle fiber membrane, which does contain voltage gated channel as in the other excitable membrane, depolarizing the membrane to the threshold and inserting a muscle cell action potential. The action potential is propagated along the muscle fiber membrane, inducing the muscle fiber contraction. FCH is in the left region is destroyed by the acetylcholine stage FCHE. Destruction is rapid before a second action potential can be initiated. Choline is taken back into the nerve terminal to be reused in the acetylcholine synthesis. फिर आता है राइट ऑन द न्यूरोमस्कुलर ट्रांसमिशन. या राइट शॉर्ट नोट ऑन द न्यूरोमस्कुलर ट्रांसमिशन. तो रेफर टू द आंसर 1 ऑफ द सेम चैप्टर. नेक्स्ट है राइट अ शॉर्ट नोट ऑन द मॉलिक्यूलर मैकेनिज्म ऑफ द मसल कॉन्ट्रैक्शन और राइट ऑन द मॉलिक्यूलर मैकेनिज्म ऑफ द स्केलेटल मसल कॉन्ट्रैक्शन. तो ये भी refer to the answer 1 of the same chapter. Write in brief about the sarcotubular system in the skeletal muscle. It is the system of the membranous structure which is in the form of the vesicle and tubules in the sarcoplasm of the muscle fiber. नेक्स्ट में फिर आता है it is the specialized system of the internal conduction. This system surrounds the myofibrils which are embedded in the sarcoplasm. The system is formed by the two types of the structure, T tubules and the L tubules. T tubules, they are also known as the transverse tubules. Transverse tubules are narrow and are formed by the invaginations of the sarcolemma. Transverse tubules penetrate from the one side of the muscle fiber to another side. T tubules open to the exterior of the muscle cell and so the ECF runs through their lumen. T tubules cause rapid transmission of the impulse in the form of the action potential from the sarcolemma to the myofibrils. फिर आता है L tubules. These are also known as the longitudinal tubules. Longitudinal tubules are the closed tubules which run off to the long axis of the muscle fiber, forming the sarcoplasmic reticulum. They form a closed tubular system around each myofibril and do not open exterior to the muscle cell. Till complete length of the myofibrils at each regular intervals, L tubules dilate to form a pair of the lateral sacs known as the terminal cisternae. Each pair of the terminal cisternae lies in close contact with the T tubule. T tubule along with the cisternae on both sides is known as the triad of the skeletal muscle. L tubule store large quantity of the calcium ions. When the action potential reaches at the cisternae of the L tubule, calcium ions are released in the sarcoplasm. Calcium ions trigger the excitation contacting coupling. फिर आता है describe the ionic basis of the action potential. तो following is the ionic basis of the action potential. Resting membrane potential during the rest. Inside of the membrane is negative and outside is positive. Ion imbalance is modified by the sodium potassium pump and selective permeability of the cell membrane. Since permeability of the K plus ion is greater than the Na plus ion, therefore the K plus channel maintains the rifting membrane potential. फिर आता है depolarization. तो at the time of the stimulation, flat decrease in the rifting membrane potential because of the passive distribution of the ion caused the increase in the K plus and the colloid in ion influx, which restore the rifting membrane potential. When the depolarization exceeds voltage of the 7 millivolt, sodium channels get activated through the M gates, that means the voltage gated sodium channel opens at increased rate, and when the firing level is reached, there is influx of the sodium along with its concentration, and electrical gradient is so high that it overcomes the repolarizing forces and run away depolarization starts. The membrane potential fails to reach the plus 50 millivolt during the action potential because increase in the sodium permeability and is for the short duration. यहाँ पर दिखा हुआ है रिफ्टिंग मेम्ब्रेन पोटेंशियल पैसिवली ICF, ECF, Na plus K plus pump. यहाँ पर दिखा depolarization reversals of the polarity Na plus influx, M plus gate और N plus Na plus channel voltage gated. फिर आता है repolarization अर्ली दिखाया हुआ है. इसमें आ गया K प्लस चैनल वोल्टेज गेटेड रैपिड K प्लस इन्फ्लक्स डिक्रीज इन द Na प्लस इन्फ्लक्स. फिर आता है आफ्टर डिपोलाइजेशन लेट स्लो डिपोलाइजेशन H प्लस गेट स्लो K प्लस रिफ्लेक्स और Na प्लस दिखाया हुआ है. फिर आता है आफ्टर हाइपरपोलाइजेशन Na प्लस H प्लस गेट स्लो प्रोलॉन्गेट K प्लस रिफ्लेक्स आयनिक बेसिस ऑफ द एक्शन पोटेंशियल. फिर आता है repolarization. तो repolarization occurs as there is a K plus influx due to the opening of the voltage gated K plus channel and there is decrease in the further Na plus influx. Opening of the voltage gated K plus channel is slow and is more for more duration than the opening of the sodium channel. The K plus influx leads to the net transfer of the positive charge out of the cell, which serves to complete the repolarization. After depolarization, at the termination of the spike potential, K plus conduction gets slowed, and few milliseconds are delayed in the restoring the membrane potential. This last phase of the flow, K plus influx, is known as after depolarization. After hyperpolarization, there is disappearance or after the depolarization, and the rifting membrane potential is achieved, but the rifting ionic status is not established. Rifting ionic status is achieved by the flow return of the K plus channel to closed gate. Write short note on the muscle spindle. Write very short answer on the muscle spindle. The muscle spindle are sensory receptors within the belly of the muscle that primarily detect the change in the length of this muscle. They convey the length information to the central nervous system by the sensory neuron. This information can be processed by the brain to determine the position of the body parts. The response of the muscle spindle to change in the length also play an important role in regulating the contractions of the muscle by activating the motor neurons by the stretch reflex to resist the muscle stretch. Muscle spindles are found within the belly of the muscle embedded in the extrafusal muscle fibers. Muscle spindles are composed of 3 to 12 interfusal muscle fibers, of which there are 4 types: dynamic regular bag fibers, bag 1 fibers, static nuclear bag fibers, bag 2 fibers, nuclear chain fiber, axon of the sensory neurons. Axons of the gamma motor neurons also terminate in the muscle spindle. They make synapse at the either or both of the ends of the interfusal muscle fiber and regulate the sensitivity of the muscle sensory afferents, which are located in the non-contractile central equatorial region. Muscle spindles are encapsulated by the connective tissue and are aligned parallel to the extrafusal muscle fiber, unlike the Golgi tendon organ, which are oriented in the series. The muscle spindle has both sensory and the motor components. Next, primary and the secondary nerve fibers spiral around it and terminate on the central portions of the interfusal muscle fiber, providing the sensory components of the structure by the stretch-sensitive ion channels of the axon. In the mammals, including humans, the motor component is provided up by the dozen gamma motor neurons and to a lesser extent by 1 or 2 beta motor neurons. Gamma and the beta motor neurons are called the fusimotor neurons because they activate the interfusal muscle fiber. Gamma motor neurons innervate only interfusal muscle fiber, whereas the beta motor neurons innervate both extrafusal and interfusal muscle fiber, and so are referred to as the skeletal fusimotor neurons. Fusimotor drive causes a contraction and a stiffening of the portions of the interfusal muscle fiber. फिर आता है right in brief on the contractile potence of the muscle. So these are also known as the contractile elements of the muscle. Myosin filaments are formed by the protein molecule known as the myosin molecule. Actin filaments are formed by the 3 types of the protein known as the actin, tropomyosin, and the troponin. All these 4 proteins constitute the contractile प्रोटीन ऑफ द मसल। फिर आता है मायोसिन मॉलिक्यूल। तो मॉलिक्यूलर वेट ऑफ द मायोसिन मॉलिक्यूल इज 4,80,000। इट कंसिस्ट ऑफ द 6 polypeptide chain, 2 heavy chains and a 4 light chain. 2 heavy chains twisted around each other to form the double helix which constitute the tail and the body of the myosin molecule. Light chain combine to the terminal part of the heavy chain and form the globular head of the myosin molecule. Myosin molecule present in the skeletal muscle consists of the two heads and is known as the myosin 2. At the time of the muscle contraction, the head forms crossing bridging. नेक्स्ट फिर आता है कि एक्टिन मॉलिक्यूल। तो देर आर दीज आर द मेजर कॉन्स्टिट्यूएंट्स ऑफ द थिन एक्टिन फिलामेंट्स। देर आर अबाउट 300 से 400 एक्टिन मॉलिक्यूल इन द ईच थिन फिलामेंट। ईच एक्टिन मॉलिक्यूल इज नोन एज द एफ एक्टिन एंड derived from the G actin. Actin molecule in the actin filaments are arranged in the form of the double helix. Each F actin molecule has an active site to which myosin head is attached. फिर आता है tropomyosin. तो there are about 40 to the 60 tropomyosin molecules situated along the double helix of strand of the actin filament. Tropomyosin molecules are long filaments which lie in the groove between the two chains of the actin filament. Tropomyosin cover binding site of the actin where the myosin head come in contact with the actin. Tropomyosin act as a regulatory protein which prevents the interaction between the actin and the myosin filaments. फिर आता है troponin. They are the small globular units which are located at the intervals along the tropomyosin molecule. They have three subunits. Troponin T, it binds other troponin components to the tropomyosin. Troponin 1, it prevents the interaction of the myosin head with the active site on the actin. Troponin C, it consists of the binding site for the calcium ions which starts the muscle contraction. How many muscles are present in our body? Compare and contrast them. There are three types of the muscle present in our body: a skeletal, cardiac and the smooth muscle. अब यहाँ पर दिया हुआ है comparison of the muscle present in our body. तो location of skeletal muscle, most of them are attached to the skeleton. Card or structure to show well-developed cross of striation, non-symphytial, lack the anatomic and the functional connection between individual muscle fiber. Control under the voluntary control. Size and the shape, it is 1 to 40 millimeter long and 50 to 100 micrometer in diameter. It is cylindrical. Sarcoplasmic reticulum, तो it is well-developed or sarcotubular system present. T system at the AI junction, terminal system prominent. फिर आता है कार्डियक मसल। तो लोकेशन इन द हार्ट और स्ट्रक्चर कंसिस्ट ऑफ द वेल डेवलप्ड क्रॉस ऑफ स्ट्रेशन एंड फंक्शनली syncytial. फिर कंट्रोल तो involuntary और size and the shape, it is 100 micrometer long and 15 micrometer long. It is short and cylindrical. Sarcoplasmic reticulum, it is more developed than the skeletal muscle. Sarcotubular system, it is present with the purely developed terminal system. The T system is prominent and is present at the jet lines. फिर आता है smooth muscle। तो location in the hollow viscera. A structure lacks cross of striation. There are two types, that means single unit which is functionally syncytial and multi-unit which is functionally non-syncytial. फिर control involuntary or size and the shape, it has variable size and is elongated. Sarcoplasmic reticulum, तो it is purely developed और sarcotubular, तो it is present but not so characteristic. इसके बाद फिर आता है nerve supply। तो और skeletal muscle में देख रहे हैं तो बायोसोमैटिक नर्व एंड बायोफेफल nerve endings. Control and the rhythmicity, it does not normally contract in the absence of the nervous stimulation. Under voluntary control, so it is known as the voluntary muscle. Blood oxygen and oxygen consumption, it is 840 mm per minute with the moderate oxygen consumption. Resting membrane potential -90 mV और absolute refractive period 1 to 3 millisecond. फिर आता है cardiac muscle, it is by two branches of the autonomic nervous system with the ganglia and the free nerve terminalis. It contracts rhythmically and spontaneously in absence of the external innervation because of the presence of the pacemaker tissue. It is involuntary. It is 250 ml per minute with a high oxygen consumption, -80 mV resting membrane potential और absolute refractive period आता है 180 to 200 millisecond. A smooth muscle, it has two branches of the autonomic nervous system with ganglia and the free nerve terminalis. Rhythmicity is of two types, that means regular and the irregularly discharging pacemaker. It is involuntary. Blood supply in oxygen consumption, it is 350 ml per minute with the less oxygen consumption. Resting membrane potential -55 mV और absolute refractive period तो not defined. Next है define the neuromuscular junction. तो neuromuscular junction is a junction between the motor nerve and the skeletal muscle fiber. A typical neuromuscular junction is seen in the skeletal muscle fiber. Write a very short answer on the resting membrane potential. So resting membrane potential defined as the electrical potential difference across the cell membrane under the resting condition. It is also known as the membrane potential, transmembrane potential difference or the transmembrane potential gradient. When two electrodes are connected to the cathode ray oscilloscope have suitable amplifier and is placed over the surface of the muscle fiber, there should be no potential difference. That means a zero potential. But if one electrode is inserted into the interior of the muscle fiber, potential difference is seen across the sarcolemma. So there is negativity inside and positivity outside the muscle fiber. Usually this potential difference is constant and known as the resting membrane potential. Condition of the muscle during the resting membrane potential is known as the polarized state. In the human skeletal muscle, resting membrane potential is -90 mV. अब नेक्स्ट टॉपिक है डाइजेस्टिव सिस्टम। So write on the composition and the functions of the saliva. Write a short note on the functions of the saliva. Yeah, write short note on the saliva. Write on the composition and the functions of the saliva. Mention brief function of the saliva. Write briefly on the composition and the function of the saliva. Yeah, write short note on the composition and the functions of the saliva. The functions of the saliva: cleaning of the mouth. Since saliva is battery, it produces a flushing action on the teeth, which helps in removing the food debris, fiber, and non-adherent food forms for the bacteria which collects over the teeth. Lubrication and deglutition. Saliva provides the lubrication to the oral tissue by the mucus and the various other glycoproteins which help provide the lubrication at the time of the speech. It also helps in the formation of the bolus which can easily slide into the esophagus. Antimicrobial function. Saliva consists of the various components which produce the antimicrobial activity. The components are the lysozyme, lactoferrin, histidines, and the salivary peroxidase. Saliva also consists of immunoglobulins such as the IgA, which provides the antimicrobial action by the agglutination, stimulating certain microorganisms by preventing their adherence to the oral mucosa. Buffering action. Saliva consists of the bicarbonate ions which neutralize the acids which are produced by the bacteria. These acids can cause the deolution of the teeth and cause the dental carriage. So due to the buffering action, saliva dissolve the acid and prevent the carriage. Digestive function. Saliva consists of the digestive enzymes such as the amylase and the lipase. These enzymes cause a start digestion of the food from the oral cavity. Enzyme amylase and lipase break a starch into the maltase and lipids into the diglycerides and the free fatty acid. Mineralization. Tooth surface is always coated with the saliva. Since saliva consists of the calcium and the phosphate ions, they increase the surface hardness of the teeth, which provide the resistance to the teeth to demineralization. Taste. Saliva dissolve the food substance so that they can be perceived by the receptor located on in the taste bud. Tissue repair. Saliva consists of the epidermal growth factor and the vascular endothelial growth factor which lead to the repair and the regeneration of the oral tissue. Excretion. Many substances from the blood reaches the saliva and saliva is considered as the root of the excretion. Here are the composition of the saliva. The saliva contains 99% of the water, 1% of the organic and inorganic substances. Inorganic contents of the saliva, calcium and the phosphate. They protect the mineralized enamel surface from the deolution. Calcium ion concentration in the saliva is 1.5 millimole per liter. Phosphate ion concentration is 5.6 millimole per liter. Fluoride promotes the remineralization of the teeth which are decaying by the dental caries. Fluoride concentration is about 1.5 millimole per liter. Hydrogen carbonate, it acts as the buffering agent in the saliva. Concentration of the hydrogen carbonate is 2.9 millimole per liter. It neutralizes the acid released by the bacteria. Thiophanate is oxidized by the salivary peroxidase and it is converted to the hypothiophanate. Hypothiophanate acts as an antibacterial agent. Its concentration in the saliva is about 2.5 millimole per liter. Other inorganic components, sodium ions are in very less quantity in the saliva. Its concentration increases along with the increase in the flow rate. Potassium ions are the major inorganic ions in the saliva as they are secreted throughout the ductal system. Other inorganic components such as the lead, cadmium and the copper. Here are the organic contents of the saliva. So the organic contents of the saliva are as follow. Amylase, it constitutes the major components of the salivary protein. That means about 50% amylase digests the starch. Mostly salivary amylase is secreted from the parotid gland. Proline-rich proteins, they constitute about 40 to 50% of the salivary protein. They reside in the salivary pellicle. Pellicle act as a diffusion barrier and slow the attacks by the bacterial acid and the loss of the dissolved calcium and the phosphate ions. Mucus consists of a large, heavily glycosylated protein. It forms the 5 to 10% of the salivary protein. Mucus acts as a diffusion barrier against the contact with a noxious substance. It also acts as a lubricant to minimizing the fear of stressage. Pirata lingual lipase, it is secreted by the bone amylase gland as well as the parotid gland. It helps in the digestion of the milk fat in the newborns. Astatharin has very high affinity for the calcium and the phosphate minerals. They stop the precipitation of the supersaturated calcium phosphate in ductal saliva as well as in the oral fluid. Lysozyme, this enzyme play important role in the antibacterial action. Lactoferrin, it has an antibacterial property. The oxidized iron part of the lactoferrin oxidizes bacteria by the formation of the peroxides which cause the breakdown of the cell membrane. Statins, they have the antifungal property. They also disrupt the cell cycle and cause the generation of the reactive oxygen species. Other organic components, the various blood group antigens are secreted in the saliva. Sugars like the glucose are secreted in the saliva. Steroid hormones like the cortisol, estrogen, and the testosterone are secreted in the minimal small quantities. Ammonia and the urea are also present in a very less quantity. Write briefly on the functions of the stomach. The following are the functions of the stomach of storage. The food is stored in the stomach for a long period before entering the intestine. The maximum capacity of the stomach is 1.5 liter. Mechanical, the peristaltic movement of the stomach mixed with the bolus with the gastric juice and convert into the chyme. Digestive, the gastric juice is secreted by the gland of the stomach and contains the enzyme which act on the protein. Protective, the HCl present in the gastric juice destroys many bacteria entering the food body along the food. Hemopoietic, the intrinsic factor of the caffeine present in the gastric juice is necessary for the absorption for the vitamin B12, which is called intrinsic factor. Vitamin B12 is an important maturation factor during the erythropoiesis. Pirata excretory, many substances like the toxins, alkaloids, and the metals are excreted through the gastric juice. Next, describe the composition, function of the various important components and the control of the secretions of the gastric juice. So, write briefly on the function and the composition of the gastric juice. Describe the composition and the functions of the various important components and secretions of the gastric juice. Describe the composition and the functions of the gastric juice. Give mechanism and the regulation of the gastric secretion. Write short note on the regulation of the gastric juice. Write short note on the functions of the gastric juice. Write about the composition and the functions of the gastric juice. Write on the composition, mechanism and secretions of the gastric juice and its function. तो कंपोजिशन ऑफ द गैस्ट्रिक जूस में आता है इट कंटेन्स 99.5% of the butter and 0.5% of the fluid. The fluid are organic and inorganic substance. The organic substance in the gastric juice are the gastric enzyme. The enzyme present in the gastric juice are the pepsin. It is main proteolytic enzyme in the gastric juice. Renin, it is milk curdling enzyme and is not present in the man. Gastric lipase, it is a big lipolytic enzyme. Other gastric enzyme, the other enzymes of the gastric juice are the gelatinase and the urease. Gastric mucus, it is secreted by the neck cells of the gastric gland. It is of the flexural gel covering the gastric mucous membrane. Intrinsic factor, this is necessary for the absorption of the intrinsic factor. The inorganic substance in the gastric juice are the HCl, sodium, calcium, potassium, chloride, bicarbonate, phosphate, and the sulfate. Functions of the gastric juice, digestive function. The gastric juice acts on the proteins and enzymes acting on the proteins are the pepsin, renin, and the gastric lipase. Pepsin, it is the main proteolytic enzyme in the gastric juice. It converts the proteins into the proteases, peptones, and the polypeptides. Renin, it is a milk curdling enzyme present in the animals and not in the man. फिर आता है gastric lipase. It is the big lipolytic enzyme. It converts the lipid into the fatty acid and the glycerol. Hemopoietic function, the intrinsic factor present in the gastric juice play an important role in the erythropoiesis. It is necessary for the absorption for the intrinsic factor from the GI into the blood. Protective function, the mucus present in the gastric juice is responsible for the protection of the wall of the stomach. The functions of the mucus are the mucus lubricates the gastric mucosa and protects it from the mechanical injury, or it prevents the digestive functions actions of the pepsin on the gastric mucosa. Actions of the HCl present in the gastric juice has the flying function, activates the pepsinogen to the pepsin. It has the bacteriolytic action, provides the acidic medium for the functions of the enzyme. Next, a mechanism of the secretions of the gastric juice. The secretions of the pepsinogen. Pepsinogen is synthesized from the amino acid. The pepsinogen molecule are packed into the zymogen granules by the Golgi apparatus. When the zymogen granules is secreted into the stomach from the chief cell, the granule is dissolved and pepsinogen is released into the gastric juice. Pepsinogen is activated into the pepsin by the HCl. Secretions of the HCl. HCl secretion is an active process taking place in the canaculi of the parietal cell. The energy is derived from the oxidation of the glucose. In the parietal cell, all the CO2 combine with H2O to form the carbonic acid. Carbon dioxide is derived from the metabolic activity in the cell. The carbonic acid is formed in the presence of the carbonic anhydrase. It is present in the high concentration in the parietal cell. Carbonic acid is the most unstable compound. Immediately, it splits into hydrogen and the bicarbonate ion. The hydrogen ion is actively pumped into the canaculi of the parietal cell. Simultaneously, the chloride ion is also pumped into the canaculi actively. The chloride is derived from the sodium chloride of the blood. Now the hydrogen ion combines with the chloride ion to form the hydrochloric acid. To compensate the loss of the chloride ion, the bicarbonate ion from the parietal cell enters the blood and combines with the sodium to form the sodium bicarbonate. Next, regulations of the gastric secretion. So the regulations of the gastric secretion occurs in the three phases. Cephalic phase, this is purely under the nervous control. By taking food, the secretions of the gastric juice starts before the food enters the stomach. The impulses are sent from the head and this phase is called as the cephalic phase. This causes the gastric secretion where food is placed in the mouth. Afferent impulses arising from the taste buds and the other receptor in the mouth reach the appetite center in the amygdala and the hypothalamus. From here, efferent impulses pass through the dorsal nucleus of the vagus. The gastric secretion occurs by the release of the acetylcholine. Gastric phase, the phase in the under the nervous and the hormonal control. When the food enters the stomach, secretions of the gastric juice increases. Pepsinogen and HCl are present in a large quantity. Intestinal phase, when the chyme leaves the stomach and enters the intestine, the secretions of the gastric juice is increased and later on it is inhibited. अगला है राइट ऑन द प्रोटियोलिटिक एंजाइम ऑफ द जीआईटी. तो Enzymes of the pancreatic juice. So pancreatic juice play an important role in the digestion of the proteins and the lipids. It also digests the carbohydrate. Digestion of the protein. Digestion for the protein is carried by the following proteolytic enzyme present in the pancreatic juice: trypsin and the chymotrypsin. They are secreted as an inactive trypsinogen and the chymotrypsinogen respectively. Trypsinogen इंट्रोकाइनेज ट्रिप्सिन में और chymotrypsinogen ट्रिप्सिन से chymotrypsin में कन्वर्ट होता है. एक्टिव है दोनों. इंट्रोकाइनेज secreted by the buffer border cell or the duodenal mucous membrane. Once formed, trypsin also activates the trypsinogen by the means of the autocatalytic reaction. Trypsin and the chymotrypsin is an endopeptidase because it breaks into the interior bond of the protein molecule by the hydrolysis. तो प्रोटीन को ट्रिप्सिन कीमोट्रिप्सिन पेप्टॉन्स पॉलीपेप्टाइड्स में कन्वर्ट करेगा. फिर यहाँ पर दिए हुए पैनकैटिक जूस में बाटर होता है 99.5%, सॉलिड होता है 0.5%. ऑर्गेनिक सब्सटेंस होता है एंजाइम और अदर ऑर्गेनिक सब्सटेंस एल्बुमिन एंड ग्लोबुलिन. फिर एंजाइम्स में आता है प्रोटीओलिटिक एंजाइम में ट्रिप्सिन, कीमोट्रिप्सिन, कार्बोक्सीपेप्टाइडेज, न्यूक्लिएज, इलेफ्टेज एंड कोलेजिनेज. और इधर लिपोलाइटिक एंजाइम में आता है पैनकैटिक लैपेज, कोलेस्ट्रॉल इस्टर हाइड्रोलेज, फॉस्फोलाइपेज ए, फॉस्फोलाइपेज बी, कोलिपिटेज, बाल साल्ट एक्टिवेटेड लाइपेज. और एमोलाइटिक एंजाइम में आता है पैनकैटिक एमोलेज. अदर ऑर्गेनिक सब्सटेंस एल्बुमिन एंड ग्लोबुलिन का दिखाया हुआ है. इन ऑर्गेनिक सब्सटेंस में सोडियम, कैल्शियम, पोटेशियम, मैग्नीशियम, बाइकार्बोनेट, कोलाइड, फॉस्फेट और सल्फेट आते हैं. नेक्स्ट है न्यूक्लिएज. तो न्यूक्लिएज इज इन पैनकैटिक जूस आर रिबोन्यूक्लिएज एंड डीऑक्सोरिबोन्यूक्लिएज व्हिच आर रिस्पांसिबल फॉर द डाइजेशन ऑफ द न्यूक्लिक एसिड. दिस एंजाइम कन्वर्ट द न्यूक्लिक एसिड इनटू द मोनोन्यूक्लियोटाइड्स. न्यूक्लिक एसिड को न्यूक्लिएज है जो मोनोन्यूक्लियोटाइड्स में कन्वर्ट कर देता है. नेक्स्ट है इलेफ्टेज. तो प्रो इलेफ्टेज ट्रिप्सिन को इलेफ्टेज में कन्वर्ट करता है जो कि एक्टिव है और प्रिकर्जर होता है इलेफ्टेज डाइजेस्ट इलेफ्टिक फाइबर्स. नेक्स्ट है कोलेजिनेज. तो एक्टिवेटेड बाय द ट्रिप्सिन एंड कॉज द डाइजेशन ऑफ द कोलेजन. डाइजेशन ऑफ द लिपिड. पैनकैटिक लैपेज इट इज a powerful lipolytic enzyme. It hydrolyzes neutral fat like the triglyceride. It converts the triglyceride into the monoglyceride and the fatty acid. Phospholipase A. It is activated by the trypsin. It acts on the phospholipids and converts methane and cephalin to the lysolecithin and the lysocephalin. Phospholipase B. It is activated by the trypsin. It acts on the phospholipid, that means the lysolecithin and the lysocephalin, and convert them to the phosphorylcholine. Digestion of the carbohydrate. Pancreatic amylase is a single amylatic enzyme present in the pancreatic juice. It converts into the maltose. फिर आता है functions of the pancreatic juice. So digestive function. It has an important role in the digestion of the proteins and lipids and also digest the carbohydrate. Neutralizing action. When the acid chyme enters the intestine from the stomach, the pancreatic juice with more quantity of the bicarbonate ion is released into the intestine. Due to the presence of the large amount of the bicarbonate ion, the pancreatic juice is highly alkaline and neutralizes acidity of the chyme in the intestine. Regulations of the pancreatic juice. Cephalic phase. When food is taken in the mouth, there is secretions of the pancreatic juice. Slight thought of the food also cause the secretions of the pancreatic juice by the conditioner reflex. The afferent nerve impulse from the cerebral cortex reach the dorsal nucleus of the vagus. From the dorsal nucleus, afferent impulse reach the pancreas by passing through the afferent fibers of the vagus nerve. The vagal nerve fibers cause the secretions of the pancreatic juice by releasing the acetylcholine, which stimulates the afferent cell to release the enzymes. Gastric phase. When food enters the stomach, gastrin and the GIT hormone is secreted from the stomach. The gastrin is transported by the blood and while reaching the pancreas, it causes the release of the pancreatic juice. Next है intestinal phase. So when the chyme from the stomach enters the intestine, more amount of the pancreatic juice is secreted. This is due to two hormones, secretin and the cholecystokinin or the CCK. Acidic chyme enters the release of the secretin from the F cell present in the mucosa of the upper part of the small intestine. Secretin stimulates the pancreatic juice reaching the aqueous component. Presence of the product of the protein digestion such as the amino acid and the peptides, and the products of the fat digestion such as the fatty acid and the monoglycerides in the chyme evoke the pancreatic secretion. Rich in the enzyme, this is mediated by the cholecystokinin secreted from the first cell in the intestinal mucosa. Cholecystokinin potentiates effect of the secretin on the ducts and secretin potentiates effect of the cholecystokinin on the afferent cell. Next, I write a short note on the bile composition and the function. I write a short note on the functions of the bile. The compositions of the bile. Bile contains 97.6% of the water and 2.4% of the solid. The solids are organic and inorganic substances. Organic substances, bile salts, bile pigment, cholesterol, lecithin and the fatty acid. Inorganic substances, sodium, calcium, potassium, chloride and the bicarbonate. Here functions of the bile matter. Digestive function, as lipids are insoluble in the water. Various lipolytic enzymes cannot digest lipid. Lipids are insoluble in the water because of the surface tension. Due to the deterrent action, bile salts reduce the surface tension of the lipids and lipid become water soluble. This is known as the emulsification of the lipid. Absorptive function, when the bile salts combine with the lipid, mucilage are formed. The lipids of the mucilage become water soluble and are easily absorbed. This action of the bile salt is called the hydrotropic effect. Maintenance of the pH in the GIT as the bile is highly alkaline, it will neutralize the acidic chem and hence the optimum pH is maintained for the actions of the diaphragm enzyme. Prevention of the gallstone formation, the bile salts skip the lecithin and the cholesterol in the solution. In the absence of the bile salts, cholesterol precipitates along with the lecithin to form the gallstones. Antiseptic action, bile is a natural detergent, so inhibits the growth of the certain bacteria in the intestine. Laxative action, intestinal motility is stimulated by the bile salt. These actions of the bile salt help in the defecation. Chloritic action, bile stimulates liver to secrete the more bile, which lead to the fat digestion. Further, I describe in brief functions of the liver. Write short note on the functions of the liver. The following are the functions of the liver. Storage function. Many substances are stored in the liver: glycogen, amino acid, iron, folic acid, etc. Secretions of the bile. Liver secretes bile, which contains the bile salts, bile pigment, cholesterol, fatty acids, and the lecithin. The functions of the bile are mainly due to the bile salts. Next है defensive and the detoxification function. So the buffer cells of the liver play a major role in the defense mechanism. Liver is also involved in the detoxification for the foreign body. Metabolic function. Liver is an organ where the maximum metabolic actions are carried out. Metabolism of the carbohydrate, proteins, lipids, and the nutrients occur in the liver. Hemopoietic function. In this fetus, blood cells are produced in the liver. Heat production. Due to the metabolic action, heat is produced in the liver. Excretory function. Certain exogenous dyes like the bromsulfathalin and the rose bengal dye are excreted through the liver cell. Synthesis function. Liver is the site for the synthesis of the plasma protein, mainly albumin, and to the some extent alpha and the beta globulins. Liver causes the conversion of the pre-prothrombin to the active prothrombin in the presence of the vitamin K. It also secretes the other clotting factors such as the fibrinogen factor 5, 7, 9, and 10. It secretes the various enzymes such as the alkaline phosphatase, serum glutamic oxaloacetic transaminase, serum glutamic pyruvic transaminase. Liver removes the ammonia from the body to synthesize the urea. फिर आता है miscellaneous function. The liver acts as a reservoir of the food and stores the 650 ml of the blood. It also regulates the blood volume. Liver leads to the inactivation of the some hormones such as the insulin, glucagon, vasopressin. Destruction of the RBC also occurs in the liver. अगला है राइट शॉर्ट नोट ऑन द बाइल salts. The bile salts are the sodium and the potassium salts of the bile acids which are conjugated with the taurine or the glycine. Bile salts are of two types, primary and the secondary. Primary bile acids are cholic acid and the deoxycholic acid which are synthesized by the hepatocytes from the cholesterol. Secondary bile salts are the deoxycholic acid and the lithocholic acid which are formed from the primary bile salts in the colon by the actions of the intestinal bacteria. In the liver, bile acids are conjugated with the glycine or the taurine and form the conjugated bile acids. Conjugated bile acids that make glycocholic acid and the taurocholic acid form bile salts in combination with the sodium or the potassium. फिर आता functions of the bile salts. So the bile salts are required for the digestion and absorption of the fat in the intestine. Emulsification of the fat, bile salts break large fat drops into the small drops. Formations of the micelle. Bile salts combine with the products of the hydrolysis of the triglycerides and form the micelle which gets transported to the birth border of the epithelial cells for the absorption. Absorption of the fat soluble vitamin. Bile salts form the complexes which are more soluble in the butter. Laxative action. Intestinal motility is stimulated by the bile salts. This action bile salts help in the defecation. Chloritic action. Bile stimulates the liver to secrete the more bile which leads to the fat digestion. Bile salt keep the cholesterol in the soluble form in the gallbladder. Bile this prevents the formation of the gallstone. फिर यहाँ पर कोलेस्ट्रॉल है, कॉलिक एसिड, डीऑक्सीकॉलिक एसिड, कीनो डीऑक्सीकॉलिक एसिड और इधर आता है प्राइमरी बाइल साल्ट, फिर लिथोकॉलिक एसिड और सेकेंडरी बाइल साल्ट. Bile salt, colic acid plus glycine or taurine. Conjugated bile acid plus glycolic acid, Na plus or K plus. Bile salt, sodium glycochelate. In the liver में हो रहा है formation of the bile salt from the bile acid. अगला है write a short note on the intrahepatic circulation. The flow of the blood from the intestine to the liver through the portal vein is on the intrahepatic circulation. और यहाँ पर दिए हुए गॉल ब्लैडर, लिवर, पोर्टल वेसल और इंटेस्टाइन. Intrahepatic circulation है ये. Total bile salt which enter the duodenum, out of them 90 to 95% of the bile salts are deviated from the terminal ileum in the portal vein and return to the liver to excrete again. This is intrahepatic circulation. Intrahepatic circulation, bile salts are necessary due to the limited amount of the bile salts present for the digestion and absorption of the fat. फिर आता है write in brief on the deglutitions. ये write in brief on the second stage of the deglutition. Deglutition for the swallowing is process that transport a chewed food from the mouth to the stomach. It occurs in the three stages: oral stage, pharyngeal stage, or esophageal stage. तो पहला आता है ओरल स्टेज. When the chewed food is ready for the swallowing, it is voluntary rolled back into the pharynx by the backward and upward pressure of the tongue against the palate. Or pharyngeal stage. In the pharyngeal stage, bolus is pushed from the pharynx into the esophagus. It is an involuntary stage. Following are the events occurring during the movement of the bolus from the pharynx into the esophagus. Oral cavity closes from the pharynx due to the approximation of the posterior pillars of the passage. Nasopharynx is closed due to the upward movement of the soft plate. This prevents the regurgitation of the food in the nasal cavity. Plateau pharyngeal fold get pulled medially and make slit-like opening of the food for which allow properly masticated food to pass through. Vocal cord approximate, stopping the breathing temporarily. Larynx is elevated and pushed entirely by the cheek muscle, which led to the enlarging of the opening of the esophagus. Epiglottis swing backward to close the laryngeal opening. This guides the food towards the esophagus and prevents it entering the trachea. Upper esophageal sphincter normally remain contracted, open up, and allow the bolus of the food to be pushed in the upper part of the esophagus due to the peristaltic contraction wave of the pharynx, which also continues in the esophagus. As the bolus of the food passes in the esophagus, contractions of the cricopharyngeus occur, vocal cords open up, which allow normal breath to resume, and upper esophageal sphincter come in tonic contraction. This stage gets completed in the 1 to 2 seconds. फिर आता है esophageal stage. It is an involuntary stage. The functions of the esophagus is to transport the food from the pharynx to the stomach. When the bolus reaches the esophagus, the peristaltic waves are inserted which push the bolus into the stomach. Two types of the peristaltic contraction start in the esophagus: primary peristaltic contraction and the secondary peristaltic contraction. Right in brief on the gastric emptying, the slow transfer of the food from the stomach to the intestine is called as the gastric emptying. Causes the peristaltic waves rising in the pyrolytic part of the stomach or simultaneous relaxations of the pyrolytic sphincter. Next, a factor affecting the gastric emptying. The volume of the gastric content in the content of the stomach is more. A large amount is inputted into the intestine. Consistency of the gastric content. Emptying of the stomach is proportional to the consistency of the content. Water is inputted into the intestine as soon as it is swallowed. Then the liquid and the solid after being converted to the fluid. यहाँ पर टंग हो गया, बोलस हो गया, सॉफ्ट पैलेट हो गया, फिर सुपीरियर फेरेंजियल कंस्ट्रिक्टर मसल, मिडिल फेरेंजियल मसल, इपिग्लोटिड, इसोफेगस, लैरिंक्स और मिडिल एंड द इंफीरियर फेरेंजियल कंस्ट्रिक्टर मसल. फिर यहाँ पर फेजेज ऑफ द डिग्लूटेशन दिखाया हुआ है, फेरेंजियल इसोफेगल कंस्ट्रिक्टर मसल है जो दिखाया हुआ है. Chemical composition: the gastric content with more carbohydrate leaves the stomach more rapidly than content with the protein. Protein leaves the stomach more rapidly than the fat. pH of the gastric content: the strong acid content leaves the stomach slowly. The emptying of the such content is accelerated by the neutralizing acid. Osmolar concentration of the gastric content: the gastric content which is also tonic to the blood leaves the stomach rapidly. फिर आता है regulations of the gastric emptying. The emptying of the stomach stops mainly due to the inhibition of the gastric motility. The inhibition motility of the stomach is caused by the nervous factor or hormonal factor. Nervous factor में आता है the nervous factor which regulates the emptying of the stomach is intragastric reflex. और hormonal factor, the major hormone which controls the gastric emptying is the introgastron. यहाँ पर हो गया gastric emptying में हो गया stomach sphincter, circular muscle relax, previous mouth full of the food swallowed, circular muscle contact, esophagus और food दिखाया हुआ है. Next है write in brief on the movement of the small intestine. Write a short note on the propulsive fermentation movement. The movement of the small intestine. The movement of the small intestine is essential for mixing of the chyme with the digestive juice, propulsion of the food and absorption. The movement of the small intestine are mixing movement. The mixing movement of the small intestine are essential for the proper mixing of the chyme with the digestive juice like the pancreatic juice, bile and intestinal juice. The mixing movement are of two types: fermentation movement. They are common type of the movement of the small intestine which occur in the rhythmic fashion. So these movements are called the rhythmic fermentation movement. The contraction occurs at a regularly spaced intervals along the sections of the intestine. The segment of the intestine in between the contracted segment are relaxed. The length of the relaxed segment is same as that of the contracted segment. After some time, the contracted segment are relaxed and relaxed segment are contracted. Therefore, the segmentation contraction chop the chyme many times. This helps in mixing the chyme with the digestive juice. Next step, pendular movement. A small constrictive wave sweep forward and backward or upward and downward, and the intestinal loop move like a pendulum of the clock, and this is called as the pendular movement. Propulsive movement. The movement of the small intestine involved in the pushing chyme towards abnormal end of the intestine are called as the propulsive movement. They are of two types: peristaltic or peristaltic rust. The peristaltic movement. Peristalsis is defined as a wave of the contraction followed by the wave of the relaxation which travels abnormally. The stimulation of the smooth muscle of the intestine initiates the peristalsis. The peristaltic contraction starts at any part of the small intestine and travels towards an end at the velocity of 1 to 2 cm per second. The contractions are big and disappear after traveling few centimeter distance. Because of this, average movement of the chyme through the small intestine is slow and average velocity less than 1 cm per second. Thus, the chyme requires several hours to travel from the duodenum to the end of the small intestine. Peristaltic rough, sometimes a small intestine slow powerful peristaltic contraction. This is caused by the excessive irritations of the intestinal mucous membrane. This type of the powerful contraction begins in the duodenum and passes through the whole length of the small intestine and finally reaches the ileocecal wall within a few minutes. This is known as the peristaltic rough. Purata, I write a short note on the local GIT hormone. I write briefly on the gastrointestinal hormone. Three hormones produced by the GIT gastrin. Source of the secretion, it is mainly secreted by the cells of the pyloric glands and also by the G cells of the stomach, duodenum and the jejunum. A stimulant for the secretion, it is secreted from the stomach during the gastric phase of the gastric secretion and from the small intestine during the intestinal phase of the gastric secretion. Factors which cause the secretion of the gastrin are the presence of the food in the stomach, a stimulation of the fibers of the local nerve plexus in the stomach and intestine, actions of the gastrin, secretions of the gastrin juice with more pepsin and HCl. Acceleration of the movement of the stomach or growth of the muscles of the stomach. Frata secretin. Source of the secretin. This by the first ever hormone discovered and is secreted by the S cells of the duodenum, jejunum and the ileum. A stimulant for the secretin. It is first produced in its inactive form known as the prosecretin and gets converted into its active form, that means the secretin, by the acidity of the chyme. The stimulant for the release and the activation of the prosecretin is acidic chyme entering the duodenum from the stomach. Actions of the secretin, alkaline pancreatic juice protects the intestinal mucus from the acid chyme by neutralizing it. It inhibits the secretions of the gastric juice. It inhibits the motility of the stomach. Priata CCK or the cholecystokinin or the pancreogemin, source of the secretion, it is secreted by the one I cells in the mucosa of the duodenum and the jejunum. A stimulant for the secretion, the stimulant for the release of this hormone is the acidic chyme containing the digestive product of the fats and the proteins. Actions of the CCK, it leads to the secretions of the pancreatic juice which is rich in the enzyme. It causes the contractions of the gallbladder to release the bile. It potentiates effects of the secretin to secrete more alkaline pancreatic juice. It enhances the secretions of the endokinase from the duodenum. CCK inhibits the gastric motility. It enhances the motility of the large and the small intestine. It encourages the pancreatic growth. It is seen in the neurons of the brain which is involved in the regulation of the food intake. फिर आता summarize the digestions of the protein. So the food stuff containing the high protein content are the meat, milk, egg, fish and the pulses. Different types of the protein which are found in the diet are the albumin, globulin, nucleoprotein, casein, collagen, gelatin, mucin and the elastin. Proteins are absorbed in the form of the amino acid, so proteins are digested to amino acid prior to their absorption. Protein digestion starts in the stomach and ends in the intestine. Enzyme responsible for the digestion of the proteins for the proteolytic enzyme. Digestion of the protein in the stomach. Pepsin is the most important proteolytic enzyme of the gastric juice. Optimum pH for the activity of the pepsin is 2 to 3, which is provided by the HCl present in the gastric juice. फिर आता है pepsin acts on the protein and converts into the peptone, proteases and the polypeptides. Digestion in the small intestine. Enzyme present in the pancreatic juice like the trypsin, chymotrypsin, carboxypeptidase and the enzyme present in the succus intricus are the dipeptidase, tripeptidase and aminopolypeptidase acts on the protein and convert it into the amino acid. Proteins are absorbed in the form of the amino acid from the small intestine. The absorption of the amino acid is faster in the duodenum and the jejunum and slower in the ileum. Next, I write a short note on the digestion and the absorption of the carbohydrate. The digestion and the absorption of the carbohydrate in the mouth. In saliva, the salivary amylase or the ptyalin is an enzyme which helps in the digestion of the carbohydrate. In mouth, the food stays for the shorter time, so the actions of the ptyalin continue for the 1 hour after reaching of stomach. Cooked starch and in products are disaccharides, dextrins and the maltose. In the stomach, gastric juice release the enzyme known as the gastric amylase. It is a big enzyme and its action is negligible. In the small intestine, here pancreatic juice contains the pancreatic amylase which acts on the polysaccharides and disaccharides and end product is disaccharide, dextrins, maltose, maltose and the monosaccharides. In the small intestine, succus intricus is present which release the sucrose, maltose, lactase, dextrinase and the trihalase. The enzyme act on the sucrose, maltose, lactose, dextrin and the trihalase and the end product is glucose, fructose and the galactose. Write a short note on the hemolytic jaundice. In the hemolytic jaundice, the excretive function of the liver is normal, but there is excessive destruction of the RBC and thus the bilirubin level in the blood is increased and the liver cell cannot excrete the much bilirubin rapidly. In this jaundice, there is a increased level of the free bilirubin in the blood. The urobilinogen gene and the pterocobilinogen is present in the excess. In the hemolytic jaundice, the anemia is present. In this type of the jaundice, the hemoglobin level get decreased. In it, the value of the SGOT is slightly elevated. अगला है describe in brief digestion and absorption of the protein. Describe the digestion of the protein in the various part of the gastrointestinal tract. तो इसमें आता है कि डाइजेशन ऑफ द प्रोटीन डज नॉट occur in the mouth as there are no proteolytic enzyme in the saliva. Enzyme responsible for digestion of the protein are called the proteolytic enzyme. Digestion of the protein in the stomach pepsin leads to the digestion of about 10 to 15% of the protein entering the gastrointestinal tract. Pepsinogen, which is in the active form, is converted into the pepsin by the action of the HCl. Pepsin split the protein into the proteases, peptones, and the polypeptides. Optimum pH for the actions of the pepsin is 2.0. Protein digestion in the stomach is important as the protein digestion stimulates the secretions of the proteolytic enzyme of the pancreas. तो यहाँ पर दिखाया हुआ प्रोटीन गैस्ट्रिक पेप्सिन प्रोटीएजेज पेप्टोन्स फॉर लार्ज पॉलीपेप्टाइड्स फिर पैनकेटिक प्रोटीएजेज डाइपेप्टाइड्स ट्राइपेप्टाइड्स पॉलीपेप्टाइड्स ब्रॉस बॉडी एंजाइम अमीनो एसिड एंड द स्मॉल पॉलीपेप्टाइड्स इंटरसेलुलर पॉलीपेप्टाइड और अमीनो एसिड समरी ऑफ द डाइजेशन ऑफ द प्रोटीन से ये दिखा रहा है. नेक्स्ट है डाइजेशन ऑफ द प्रोटीन इन द स्मॉल इंटेस्टाइन. तो proteins in the small intestine are digested by the pancreatic proteases, burst border peptidases, and the intercellular peptidases. Pancreatic proteases lead to the digestion of the protein and they break into the dipeptides, tripeptides, and the small polypeptides, which later on get digested by the burst border peptidases. Some dipeptide as well as tripeptide get absorbed in the epithelial cells of the mucosa of the small intestine and get further digested by the intracellular enzyme into the amino acid. Burst border peptidases consist of aminopeptidases, dipeptidases, tripeptidases, nuclease, and the related enzyme. They convert the protein to the small polypeptides and the amino acid. Intracellular peptidases present in the cytosol of the epithelial cells of the small intestine. They are specific for the linkage between the various amino acids. These proteolytic enzymes within the minute digest dipeptide and tripeptide in the amino acids. Prior to absorption from the protein, end products of the protein digestion get absorbed by the luminal membrane of the epithelial cells of the small intestine, flowing in the absorption of the protein. Sodium-dependent active transport mechanism, LIBO amino acid, dipeptides and the tripeptides get absorbed by this mechanism. There are separate transporters for the absorption for the basic acidic and the neutral amino acid. In this mechanism, there occur the binding of the amino acid to the sodium ion and to the sodium ion to the carrier protein. Electrochemical gradient is created across the epithelial cells and there is a movement of the sodium ion amino acid inside the cell. Simple diffusion, dextro amino acid get absorbed by this mechanism. Prior to endocytosis, a small amount of the large polypeptides are absorbed by this mechanism. Prior to right short note on the peristalsis. Peristalsis is defined as the wave of the contraction followed by the wave of the relaxation, which travels in the abroad direction. A stimulation of the smooth muscle of the intestine leads to the peristalsis. Peristaltic wave travels from the point of the stimulation in the both direction. In normal condition, progress of the contraction in an oral direction is stopped quickly and the contraction gets disappeared. The contraction that travels in an arboreal direction persists. At times, a small intestine for a powerful peristaltic contraction. This is caused by the excessive irritations of the intestinal mucosa or extreme distensions of the intestine. This powerful contraction starts in the duodenum and passes through the entire length of the small intestine and reaches the ileocecal wall within few minutes. This is called the peristaltic rush or the rush waves. Peristaltic rush shifts the content of the intestine into the colon and hence it relieves the small intestine of either irritants or excessive distension. Next है right short note on the digestion and the absence of the fat, digestion of the fat. Digestion of the fat in the mouth, salivary lipids in the active in the stomach and digest 30% of the dietary triglycerides. Digestion of the fat in the stomach. Gastric lipase is a big fat splitting enzyme and act on the optimal pH of 4 to 4.5. Next है fat digestion in the stomach occurs in the exceptional circumstances when pancreatic lipase may regurgitate into the stomach from the duodenum and in the achlorhydria. Digestion of the fat in the small intestine. Pancreatic lipase and the bile salts enter the second part of the duodenum and starts the fat digestion. Pancreatic juice is alkaline and converts and adjusts the highly acidic CAM to the CAM at the slightly above 7.0, which is optimal pH for the lipase activity. Bile salts play an important role in activating the lipase and produces detergent action by hydrotropic action and lowers the surface tension. This leads to the emulsification of the fats and provides a greater surface area for the lipase enzyme to digest. Lipase act as the interface between the fat particles and the butter and successfully hydrolyze the double and the triple bonds of the triglycerides into the diglycerides and the monoglycerides and release the fatty acid. Dietary cholesterol remains in the form of the cholesterol ester and the pancreatic bile salt activated lipase and the cholesterol in stage of the succus integrus which hydrolyze the ester in the intestinal lumen and finally convert it into the cholesterol. Prior to absorptions of the fats. So monoglycerides, pancreatic electrolytes, fatty acids and the bile salts.